One of the most intriguing titles of any session at this year's EASD Congress is the one titled Cures for Diabetes? And there are three presentations in that session, and one of them is on gene therapy. And I'm delighted to welcome Fatima Bosch from Barcelona. Fatima, when we think of gene therapy, we think of principally diseases that are caused by a single variant that's gone bad, a glitch. We don't think about polygenic diseases like diabetes. So how can gene therapy help with either type 1 or type 2 diabetes?
Well, the gene therapy has been developed mainly for treating genetic diseases, but now I think it's mature enough to move to more complex diseases like diabetes, cardiovascular disease, or cancer. And in the case of diabetes, that we have many genes involved, then it's not as easy as for a monogenic disorder in which you can take the corrected copy of the mutated gene and introduce it back to the patient. In this case, what we need is to know better the molecular mechanisms to lead to the disease and trying to counteract them. For example, in the case of type 1 diabetes, there are several approaches run by different groups in which you can engineer tissues to increase glucose uptake, to reduce hyperglycemia. For example, in our case, we have created a glucose sensor in the muscle of animal models, and we have demonstrated safety and long-term efficacy in dogs, diabetic, for up to 8 years. That means that
That's astonishing.
these animals, they didn't receive any insulin treatment, and they didn't have any beta cell in the pancreas, and they survived very long, and they were normal. That means that we can have some approaches that may reverse the disease, or they can treat the disease, but we cannot say cure. I think cure is something we can treat with gene therapy, but it will be difficult to cure, but we can treat. Other approaches can be to engineer the pancreas in vivo. There are groups working on that. We are also engineering the pancreas. You can use vectors to introduce genes that induce beta cell replication, or also genes that can induce more immunotolerance. Well, you can counteract the disease, and maybe in this case, maybe you can cure if you treat patients at early stages of the development of the immune response against the beta cells. But this is for type 1. But more difficult, I think, is for type 2, because, as you said, there are many different phenotypes, and there are already available many drugs, but we have to find treatments that can counteract the disease in patients that they have any other options. There is no live intervention, no drugs working on them. And in this case, we and other groups, what we are doing is try to find possible therapeutic candidates, and it's what I'm going to introduce today in my talk, is that we found a candidate that there were a lot of data preliminary using the protein, in which that if you use a vector to introduce this protein, in our case was HGF21, we were able to reduce or to counteract obesity, body weight gain, and to increase insulin sensitivity in 2 different animal models that were already obese and insulin resistant. That means there are a whole bunch of strategies going on trying to counteract the main phenotype of the disease. But we cannot cure them because we don't have a gene that we have to correct.
Now, the problem that's bedeviled all of gene therapy is the use of vectors. So the way that you introduce your altered genes into the body, is that still a major problem?
Well, it depends for the disease, because each disease you may have a specific vector. For in vivo gene transfer, the vectors that are now more available are the adeno-associated viral vectors derived from adeno-associated viruses that they are not pathogenic, they are very safe. But it depends of if you need to introduce a gene in the whole cells in the body, then this is very difficult, and you cannot with a vector do that. But if you need to engineer a tissue to secrete a protein, then this is much easier. And for example, nowadays you can engineer very safely, and there is data already in clinical trials that you can engineer the skeletal muscle or the liver to secrete proteins to contract diseases. So, well, there are other targets. For example, is adipose tissue also is a very good target organ to secrete proteins. It depends of what is your approach, that you select the vector. Usually it's AAV for in vivo gene therapy, but then there are several serotypes of AAV that have different cell tropism, and then you can select the best one for your approach, together with the candidate gene that you select a specific gene vector, you have the candidate gene, then you can introduce the promoters or regulatory sequences to make more strong the level of expression. And with that, you can engineer the target organ that you think is the more appropriate or easily accessible. It will depend on your approach, but you have today a whole collection of vectors, regulatory elements that you can combine. It's like to do, I would say, I don't know, you can work on more biotech, like bricolage.
So you have a toolbox
yes,
that you can pick your tool for the job in hand.
And depending on what you want, but nowadays you can engineer anything. Yeah, we are also working on the brain, and we can deliver genes very easily to the CSF to engineer the brain. And then you can combine all these with the route of administration also. This is important. I think the field has developed a lot, and now is the time for diabetes. I am completely convinced.
And that's fascinating that you think it's the time. So, and what is the time frame? Are we still talking 5 to 10 years?
Probably. We need to also, identify which is the best gene therapy that could work in humans, and then look at around the population of diabetic patients and select those that will be better candidates for this kind of therapy. Because I don't think it will be one gene therapy for all diabetes.
No, it will be very selected patients.
Yes, select a group of patients that can benefit from one type or another type of gene therapy approaches.
Fascinating. Well, thank you very much indeed. Well, what an extraordinary glimpse into the future there. We'll have more on future diabetes cures, perhaps, in a while. And you can see Fatima Bosch's presentation in full on EASD.org.